Hello. Thank you very much, Ava and the ARDD organizers, and thank you to Jamie Justice for giving me the motivation to talk today, not about longevity medicine, but about N-of-1 medicine. So I think all of us can agree in the audience today that our understanding of human biology is woefully inadequate. Worms, fruit flies, mice, fine. But if we think about human biology, it really isn't up to snuff yet. And what this is leading to in the clinical sense, where I practice, is that we have this lengthy trajectory of trial and error when we're taking care of patients. So this is kind of the classic example of what happens when a physician is seeing a patient. You have a diagnosis, you think of an intervention, the patient goes home, they try it on their own, then you wait, they come back, you might change the dose if it's not working, wait more, change the dose, and then if it's really not working, you start over with a new intervention. But this can take years. So this is very frustrating, obviously, for both the patient and the providers. And I think a lot of why this happens, why this kind of, what we call evidence-based medicine happens, is because we're still stuck in the randomized control trial world. And this is a quote from a recent N-of-1 medicine article. And I'm obviously proselytizing on N-of-1 medicine, but I just love this. The most valid and relevant evidence for a single person receiving care is unlikely to be a directly applicable group-based effect estimate from a randomized control trial or a recommendation from a guideline, since such recommendations forego an individual's personal context and circumstances. And that's just a very long way of saying no one is average. And I think what really can send this point home is if we look at the heterogeneity of response to weight management medications, which is a huge part of my practice right now. And the two on the top right here are oral medications, Contrave and Qsymia, and then the others are the new generation of the GLP-1 receptor agonists, or a dual agonist, or with the final one, retatrutide, that's a triple agonist now. And if you look, this is kind of an amalgamation of the data from these published studies. You can see that the response in weight change, and that's by percent, is completely variable within these groups. So a lot of people actually gain weight when they're given the medication Contrave. And the weight change is very variable. Even going down to the retatrutide, you can lose up to 50% of your body weight, which is enormous, but many people won't be losing that much. So I think this begs for a new approach to how we do clinical trials, but also how we practice medicine. And that's where I believe N-of-1 medicine comes in. We're all snowflakes, in a way. So why N-of-1 and not precision, personalized, or individualized medicine? Well, I think N-of-1 really invokes clinical trials. And a lot of what I do in my practice is try to stress the importance of applying scientific research principles to standard patient care. Because I think we're missing an opportunity in a lot of ways when we do clinical care to use some of the techniques that we use in clinical trials. And I've always asked why clinical trials use really complex survey instruments or bioassays or blood work, but these kinds of tools are never reproduced in the clinical setting, yet they're so rich with information that we can use to help our patients. So N-of-1 medicine is distinct from N-of-1 clinical trials, but it incorporates a lot of the elements of N-of-1 clinical trials. And I don't have time to go through all of this, but I'll start to give you a little bit of an idea what I'm talking about. So some of the basic principles of these N-of-1 trials that I think can be applied to patient care start off with longitudinal biomonitoring, and I'll go into a little bit more detail about that later. But you can also have an N-of-1 trial that you're randomizing the order of drugs and interventions. So a lot of people think, oh, if you're only looking at one person, you're not going to be able to randomize. Well, that actually isn't true. We do randomization a lot. You can also use placebos. So a lot of people also think, well, if you're just taking one person and you're only giving them one drug, you don't really have a way to counteract or understand what's happening when they're not on that drug. Well, we use placebos a lot with N-of-1 medicine. You can use blinding, too. And I think one of the most important things that I get out of this N-of-1 medicine is that you're analyzing the data. You're not just having the patient go home, try different things themselves, and come back and ask, so how'd it go? You're actually looking at the data that they're collecting on their own. And then you're doing statistical analyses on that data. So a lot of different statistical analyses that I am not smart enough to understand, but my partner, who is Dr. Nicholas Shork, is a biostatistician. He'll be speaking later. He helped me with the slide, I admit. But it's very important to actually get this data and then analyze it. And N of 1 medicine, I have to say, is not for every condition. So we have certain conditions where we really think it's appropriate and helpful, and those are chronic conditions where you have a relatively stable disease course and already approved treatments. You can also do it in rare diseases where you'd have no idea what the treatments are yet. And I also think that geriatric medicine is a great place for N of 1 medicine because elderly are often excluded from standardized randomized control trials, so you don't have a lot of good data as to how certain medications are going to respond in the elderly. And then, very important for longevity medicine, this approach is really helpful for health optimization. So any of the interventions that we've been hearing about, caloric restriction, diet, exercise, all those things, you can easily use those in the N of 1 medicine approach. So I'll just go through a little bit of the step-by-step process. As I said, I don't have time to go through each of these. But as I said, the first step is really understanding what conditions can lend itself to the N of 1 approach, and do you have that condition in the patient in front of you? And then you have to determine the intervention comparator groups. So are you going to use different medications, comparing different medications for the same condition, or different doses of the medication? Are you going to use a placebo? Are you going to use non-drug interventions like biofeedback and behavioral therapy? N of 1 has been used for a long time in behavioral therapy. So this is where we're getting a lot of our information from these psychiatric and behavioral therapy trials. Are you going to use washout periods in between? So if you're doing crossover trials, you may know that you might have effects that carry over. So washout periods might be important depending on what you're using. And then timing. Is this something that you can do in weeks, months? How long is this timing going to be? Then you have to determine what the longitudinal monitoring requirements are for the trial, for the individual that's in front of you. Are you going to be using just blood-based assays, survey instruments, biomorphic, biometric data, wearables? All these things can be used in these conditions. And then are you going to randomize the order of interventions? Then you have to worry about data collection, data analysis. And then finally, what you're going to be doing with all this information is finally having this shared clinical decision-making experience between providers and patients. And patients are very receptive to this because they really feel that they're part of their care, more so than the standard approach that we talked about. So I'm just going to talk very briefly about basic N of 1 trial designs. This is an article that Dr. Shork and I wrote, Exploring Human Biology with N of 1 Clinical Trials. And if you look on the far left, that's the most basic trial design, which is what we call an interrupted time series, where you just have somebody who's either on treatment at the beginning or and off treatment, or vice versa. You can also do reversal designs where you have more periods where they're on and off treatments. And then the final panel C shows the washout periods in green where you're actually doing a washout period in between. And I want to stress, as I said, some of the concepts in that step-by-step process, and this is the idea of the longitudinal monitoring. And this actually gets at what Jim Kirkland was talking about earlier, where you're trying to identify a causal relationship between what you're doing and the physiology of the patient. So say you're doing this with a geroprotector, one of Near's geroprotectors, and you want to measure not only biomarkers of aging, but you also want to see if this impacts a clinical phenotype, which is the VO2 max. So the biomarker of aging would be in the gray, the VO2 max would be in the red, and you're using different doses of the geroprotector. And what's important with the longitudinal biomonitoring is that you're giving this a temporal context. So while you're doing the data analysis, you're incorporating the time periods. And if you can see with the gray, there's a peak in the gray here, and then there's a lag, and then there's a peak in the VO2 max. So the biomarker of aging goes up, and then the VO2 max goes up. And this is accounted for when you're doing your data analysis, because if you didn't have that temporal context and you were just measuring contemporaneous data and you're just doing cross-sectional input, this is what your data analysis would look like. But when you get that temporal context, you're going to have a much more clean analysis, and you're going to see that linear correlation between an increase in a biomarker and then a lag time, and then you're going to see an increase in the VO2 max. Again, this is a hypothetical. This is designed by Dr. Nicholas Shork. Did I get this right, Dr. Shork? I just want to make sure. I did. Okay, good. Thumbs up. Great. So again, this is a very key concept when you're doing these longitudinal biomonitoring studies. So what does that end up looking like when you're doing them ongoing? So you have a baseline period of assessment, and then the longitudinal biomonitoring starts, and then you might have different drugs that you're giving in random order, and then you end up getting the baseline measurement at the top, and then you get the different drugs response at the bottom, and then you end up doing the modeling fit, and then the correlated variables will show up, and then you might even have an uncorrelated variable that didn't change at all during this process. So you can do this. This is kind of what the data look like after you're having the patients do these different trials, and you can really see easily what works and what doesn't. You can also have washout periods. So a lot of what we have done has already been figured out with different healthcare systems. So we're actually working with people at Cedars-Sinai in Southern California and also research institutes. And what we've done with these clinical trials protocol designs is actually stressed this integrating of the trial pieces that I mentioned earlier into routine clinical care. So that's one of the things I'm really passionate about, is making all of these great resources available to patients in the clinic. And this is one study I'll just go over really quickly. This was a woman who had a sleep disorder and also had seasonal affective disorder and depression. She was on multiple interventions at once, polypharmacy. And what she did was she took several different options to keep herself on all the medication, take herself off different ones. And what this shows is the percent of time that she was in deep sleep, and then the different interventions that she had ongoing. And what happened was that when she was on the interventions, she slept the worst. And when she was off all the interventions, she slept the best. So her psychiatrist had put her on all these and was very happy when we actually helped the psychiatrist understand that off all interventions was much better. And these N-of-1 global medicine efforts are really global. So there are a lot of different places around the world that are working on this, many more I'm sure that I haven't represented. And there's a lot of really interesting recent research that has been highlighting these N-of-1 trials, and N-of-1 medicine. But there are a lot of barriers too. And one of the groups in the Netherlands published this was actually a survey of the providers that had taken part in these N-of-1 medicine programs at their institution, and they had a lot of things that they brought up that were pretty difficult to overcome, a lot of problems with insurance providers and things like that. I won't go over all of them. But I think we have to really acknowledge that there are a lot of barriers to implementing this in the clinic, and one of the biggest things is this database and data management issues, because I think we are not able to use the standard EMR for what I'm talking about. We really need to have a merging of a standard EMR and maybe something like what we use in clinical trials in the US, REDCap, but something that can actually handle all these intense longitudinal biomonitoring and data analysis types. We want to use more remote biomonitoring platforms, include wearables, and then obviously the data processing and integration is really critical. So we need to follow interoperability standards between these different software programs, security and compliance. Obviously that's a huge issue in the US right now and probably around the world. And then when we get the data, we want to make sure that we analyze it and handle it and then transform it into a clinical context. So we are in the process right now of developing what we're calling an N-of-1 development app, N-of-1 medicine app, and we're working with a company in Santa Monica called Sidebench. We're really interested in the bidirectional data management. As I mentioned, we want patient engagement to be front and foremost, and obviously the interoperability is very important. So just to summarize, I think that N-of-1 medicine, the way I envision it, will take aspects of N-of-1 clinical trials, and we will incorporate them into clinical care. And by doing so, we're really going to improve our ability to take care of patients by expanding our understanding of this complexity of the human biology and all the heterogeneity that we're missing out on. But scaling and widespread adoption is going to face a lot of hurdles and barriers that are really similar to many of the other necessary healthcare reforms. Thank you very much.